All right, guys, I think we're going here. Okay. All right, so main thing is I got the right in front of me here. I got the practice final for bio. I also have the real final for bio right here. I'll take any questions about the practice final. That's mainly what we can do here, and just go over it, and that's it. So we're starting with Michael, who kicked us off, number 34, 35, and 37. I also put it on Google Classroom. So if you want to look on Classroom, if you don't have a copy, you can look on Classroom and get a copy. And let me share my screen with you guys so you can see what I'm looking at. There we go. Got two things here. We got this guy. That's a practice final. Again, that's on Classroom. So you can look at that. Get your own copy if you want. And we can get rolling with Michael's question, 35, 37, I think he said. All about peas, pea plants. Pea plants and Punnett squares. I'm sure that's all it is. All right. 35. If a pea plant's alleles for heights are TT, what is true of its parents? Okay, so I'm going to need to keep for this so I can draw out the Punnett square. Number 35. Okay, so we got a plant that is TT, and we have to basically reverse engineer what its parents would have been. So T, and I encourage you guys to draw along with me, either on your handout or on a piece of paper, just something, just so you can follow along with what we're doing here. So what is true of its parents? Okay, so if it's TT, that means Punnett square, it's got to make at least one kid. It's TT. The other kids could be whatever, really. So the parents have at least one little t and one little t. Right, so what does that mean? Now, the parents don't have to be tall or short. They could be either. If like you can have one parent that's short, like little t, you have one parent that's tall, big T, little t, that's fine. Like you can still get an offspring with little t, little t. What the parents both need to do is give you that little t. That little t represents the recessive allele. So it's got to be C. And I'm not 100% sure what the answers are for these. Let me look, make sure I know what I'm talking about here. Where is it? There it is. 35 is indeed C. Yep. 36, similar question, in the same vein. A tall plant, big T is crossed with a short plant, little t. If the F1 pea plants are allowed to self-pollinate, what's going to happen? So what they're saying in this one is you have two. For 36, we got big T, crossing with little t. So if you take the family that's made and allow them to cross over. So what do you make here? You make big T, little t. That's the whole family right there. They're all big T, little t. They're all heterozygous. And they're all tall, but they're all heterozygous. So if you let these individuals cross-pollinate or self-pollinate, if you take the big T, little t's and let them cross, like pollinate each other or pollinate themselves, what are you going to get? That's what the question's asking. The 36, the answer is D. Let's see why. You take that individual, and you basically, it's saying self-pollinate, so you got to cross it with itself. Let me make some room up here. So let's take this guy, bring it up here. And if you guys got questions, ask them. And once I'm done here, I'll take your other questions. And we are going to cross a big T with little t. Again, self-pollination. So it's crossing itself, same thing. So what do you got? You got one big T, homozygous dominant. You got one big T, little t, heterozygous. You got one big T, little t, heterozygous, and then you got little t, little t. So what is the answer? The offspring can be tall or short. And yeah, that's true. We can see it because we got tall, 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 and then we got one little shorty here. Hope that helps you out, Michael. Let's see what we got up here next. Okay. Oh, you said 37 too. All right, let me do 37 really quick. And then I'll get to 41, 44, and 45. Callie says she can't hear anything. Can you guys hear me? My thing says, I'm assuming you guys can hear me. It's saying it's picking me up. Respond if you guys can hear me. Like, say, type you can hear me. All right, let me do 37. I'll come back and check to see if you guys can actually hear me. Yeah. All right, good. Thank you. All right, so 37 really quick, then I'll get to Jeff's questions. This was the last one Michael was asking about. Heterozygous tall pea plant is crossed with a short plant. So heterozygous tall, we just saw what that looks like. That's big T, little t. That's heterozygous tall. It's crossed with a short plant. Short plant must be little t, little t. The probability that an F1 plant will be tall is what? So F1 just means like this first set of offspring. So when they say F1, they mean like these guys right here. F1. So what are the chances that these guys are going to be tall? So let's see here. We got big T, little t. We got big T, little t here. We got little t, little t, little t, little t. According to the handout, it says 37 is B, and that makes sense when we look at it because half of these guys are tall and half of these guys are short. So that makes sense. Our Jeff was asking about another one. Jeff was asking about 41, 44, 45. That's next. And then if you guys got any other questions, I'll be happy to take them. If you got any questions about the test, just a little insight here. It's a 70-question test. I think you guys will do really well on it. It'll be a piece of cake, especially if you go over this practice test here, especially if you watch this video. Our 41 first. Color blindness is a sex-linked trait and is more common in males than in females because the allele for color blindness is which of the following? So the answer here is C, recessive and located on the X chromosome. Let's see why. And I think I explained this in class, but some people, I'm sure some people missed it. So it's probably a good thing to go over. It's more common in males than females because the allele for color blindness is blah. So first off, it's more common in males right off the bat. Females oftentimes, so XX as a female, a woman will oftentimes not be colorblind. So we'll use, let's use R, color blindness. I don't know why, but I want to do something that's, uppercase, lowercase. We're very different. So here's a mom. She's big R, little r. It's we're talking about color blindness here, which is sex linked, which means that R is ugly, man. Let's fix that. There we go. It's sex linked, which means it's linked to the sex chromosome. So we have to deal with X's and Y's here. We'll say this dad is, I think the way I did it in class, we'll say he's normal. or he's not colorblind. So we can see the mom's a carrier, and the dad, my bad, this is a mistake on my part, the dad would carry on his X chromosome. All right, and we'll see why that works in a second. And I'll go back into the Y chromosome, and I'll show you why that's not the answer in a second too. So this is the right way you would set this up to figure it out. Working this out, so you got big X, big R, big X, big R. It's a very easy Punnett square. Then you got big X, X. We got big R and little r. So we can see right here that none of the women, like the women, will not be colorblind. Colorblindness in women is very, very rare. Very rare. I've only met one person my whole life that's a woman that's colorblind, and it's the student I had this year. So for the guy, if it's a boy, he'll be XY. This guy's big R, and this guy's going to be X little r, Y. So the color blindness gene is carried in the X chromosome, not on the Y chromosome. The Y chromosome does very few things. It basically just determines that you're going to be male, and that's basically it. The tiny little chromosome. X chromosome does a lot of stuff. It determines a lot of stuff. And one of the things is color blindness. And we can see here that it's basically only a boy would be colorblind in this scenario. So if we were to do this with a boy who, or if we were to, let's say you guess that it's on the X chromosome, it's recessive on the X chromosome. Let's see if that works out really quick, just as a, so it's color blindness. So we'll say little r or big R, I guess. I guess it could be either, really. So there's a mom, and we'll say she carries it. And it's more likely, it's probably a lot more likely the dad's is going to be regular vision. So let's do that one first and see how that works out really quick. So big X, we got big R, big X, big X, big X, big R, little r. In reality, if you're going to have it on the Y chromosome, you don't even need it on the X chromosome. Now that I'm looking at it, like you can just have it on only the Y chromosome. Like it's not even necessary to have it on the X chromosome. So let me fix all that. Because if you're going to guess that it's on the Y, it's only going to be on the Y. Anyway, here's the layout. Big R, big R. So according to this, it's very common in males, not even possible in females. So maybe if they mention that it's never occurs in females, maybe you could say, yeah, it's on the Y chromosome, but it's just saying it's more common in males, not that it never occurs in females. The other ones that Jeff was asking about was, I think 44 and 45. Let me check really quick which ones he mentioned, and then I'll get to your next questions. 44, 45, yeah. All right, 44 is talking about zygotes. All right, so the somatic cells derived from a single-celled zygote divided by which process? Okay. So the answer here has got to be mitosis. Right off the bat, it's got to be mitosis. Must be. Meiosis is only for creating sex cells. That's it. Any other cellular process of growth, of cell division, anything is going to be mitosis. All right, mitosis is general cell growth. Meiosis is only for creating sex cells. It's only for making sperm and eggs or ovums. That's it. All right, that's it. There's no other option there. So if it's something that's growing, like a brand new, zygote that's just two cells and has a potential to become a human being, then it's going to be mitosis, and that zygote will not perform meiosis until it's an adolescent male or an adolescent female that's gone through puberty. That's the only time they would do that process. And let's see what you guys got next. Okay, number 12 and 8. Kylie's saying number 8. So let's get through 8 and then 12. And then if that's all you guys got, then that's it. If you got more questions, please ask them. All right, here we go. All right, 8. Similar patterns of embryological development in different but related organisms are responsible for the formation of which of the following? Sounds like homologous structures. Yeah, that's right. All right, homologous structures. Again, we compare a bat to a human. All right, these are both mammals. There's two main reasons we know that. Well, the major reason is DNA evidence. All right, we can look at the DNA of a human, look at the DNA of a bat, we can look at the DNA of a rat, and we can see that there's lots of similarities in these three things' DNA. Another one is physiological. You can take the embryos of a bat, a human, a rat, look at them very early on, and you'll see that there's tons of similarities in how they early on develop. All right. Another one is that the structures that make up their arms basically are very similar. So a bat, a human, we know what a human hand looks like. Has the 2 bones, the radius ulna, has the 4 finger bones and the thumb bone. If you look at the rat arm or a little rat arm, it's going to have very similar structures. It's going to have all those bones. A bat, it's going to have all of those bones, and that's really the main way we learned it in class. And like the bones will be different; they'll be different shape and everything, but the structures will be there. The other similarities they're just point on here is embryological development. And you can do this really quick, and you can just Google let me see if I can. I don't know if you guys will see it if I search for it. Let me try. Let me try this really quick. Human and rat embryos. And you can see the similarities and see if there's 2. This is probably a comparison side by side up on the top right. And let me make sure you guys see this. I'm not just doing it to. Yeah, you guys see it. Okay. So look at that, human and rat. You can see the similarities very early on, before 28 days. You can see the differences are very, very, very similar. Very similar. They're mammals. We're both mammals. We both have similar evolutionary characteristics, traits. So, there's your evidence right there. The next one was 12. Let's see. Oh, yes, the charts. All right, so since you guys actually are paying attention here, I'll tell you right now, this is the chart you will see on the final. It's this one right here. So just note this chart, know that what definition goes with this chart. The question is different. So the answer for 12 is stabilizing selection. So basically, as long as you know that this is stabilizing selection, you're fine. What's happening in the chart is that you're having a population. Birth mass is changing based on some type of evolution occurring. It could be any type of evolution. We don't know. But from over time, we're seeing a change in how the birth mass is decreasing, or a percentage of population's birth mass is going into a more varied range, where it used to be that most of the population had a very set birth mass, but now we're seeing these different values over time. And this could be something that happens in human populations. If you were to take a certain human population, look at it over time, maybe birth masses have become more varied now than they were, thousands of years ago. Who knows? So this is the only one you really have to know for the test. I'm just telling you right now, because you guys showed up. I appreciate that. By the way, if someone's not here, they can still catch this video when I'm done. All right, they can still catch this video. It'll be, once it's done, it's saved. That's it. Okay, 29 and 60 and 52. All right, so I'll do 29, 60, then I'll come back around 52. And again, if you guys have any other questions, I'll be happy to take them. If not, we can close it out after that. So let's do 29 and 60 first, and then we'll go label 52. 29. What is that about? Oh, yes, the cell cycle. It's easy, right? Do a project on this. And cell cycle DNA is replicated. When's the DNA replicated? So when you think of the cell cycle, you think of like there's like some type of G1 or, you don't, at some point you want to be able to draw it out. If you're going to go into biology or any type of science, you want to be able to draw it off the top of your head. You got G1, S, G2. If you are going to go into some type of science, then make a mention in the comments. Say something about that. Say what you're going to go into. And then we got mitosis or M phase, that's one of the first things we called it, is M phase, which is just mitosis. So what's going on when cell's DNA is replicated? That's S phase. S phase stands for synthesis. All right, synthesis of DNA. Synthesize DNA. Synthesis of DNA. So that's when you're doubling up the DNA for when mitosis occurs. G1 phase is basically just normal cell growth. G2 phase is preparation for M phase. All right. Let me double check. I forgot already what the other one was. Oh, G1, S, and G2, yes, they are all a part of interphase. Correct. That is right. And I'm gonna go back and label that, and then we'll do 60. And Michael wants to be a neurosurgeon. So there you go. Thank you for participating, Michael. I appreciate that. Trying to make this interactive with you guys here. So yeah, all of this, maybe pick a different color. All of this part is interphase. Interphase. All right, the other one was 60. Last one on here. So this is like kind of like a macromolecule / DNA question. So the answer is D. All right, so let's look through these why it's not. So why is it not A? It's not A because you got adenine and thymine. Those are both bases. You only have one base per nucleotide. Our nucleotides are what make up the chain of DNA. So we got like a strand. It's a double helix, and you got the little bases. So each base is like an A, T, C, or a G. These guys are nucleotides, all the A, T, C's, and G's. Like the whole chain is a nucleic acid. But the individual units themselves are nucleotides. So each nucleotide is made up of one A, T, C, or G. That's it. So it can't be A because that's two A, T. It's an A and a T. Don't need that. You definitely got the phosphate group. And then why is it called DNA? It's called DNA because you got deoxyribose. That's where it gets the name from. That's where the D in DNA comes from, deoxyribose. And then you got nucleic components like cytosine or adenine or thymine or guanine. And then the acid components is also part of this whole thing. It can't be C because same idea. We got cytosine and guanine. We got two nucleotides, not needed. And then C is saying deoxyribose, phosphate group, and polymerase. Polymerase, that's just nonsense. No. It's a distractor, so it's got to be D. Other questions? Question 52. Okay, I label 52. I got psychologists, possibly. A plastic surgeon. Interesting. Interesting. Something about Oprah sponsors? Yeah, who doesn't love Oprah? Okay, so 52 is next, and if you guys got any more, let me hit. Michael wanted me to label some stuff on number 52. I know some of you other guys are interested in medicine too. I know some of you guys that didn't mention anything. That's fine. You know, that's fine. I don't have to say anything, but just know medicine is it's a long road to hoe. It's like a lot of work. And there are plenty of other related fields like nursing, psychology, like Jeff mentioned. And a lot of people think, you're interested in medicine, want to be a doctor, done, boom. But there are plenty of other options. You don't have to just do medicine. Like nursing is a very much needed field. You can do biotechnology, which is really needed right now. And I'll be honest with you guys, I'm just distracting right now because I'm pulling up a picture of this just so I can label it really quick because I forgot a lot of these structures already. And I'll tell you, I am mentally checked out. So this is a plant flower diagram. So let me look that up really quick. Oh yeah. What we did this in class, you guys just shouted out the answers. And I just wrote them down. So if you can shout them out in the comments, do that. Please help me out here. All right. So we said A was ovum and B was ovary or something like that. Ova or ovum. I forgot what we called it. And then B was the actual ovary. Just kind of holds the ovum. You got the big structure, that's the whole female structure. C is going to be the style. Oh man, this thing, I cannot write on this thing today. Let's see if you guys are saying the answers yet. Oh, Michael, you want to be a nurse? That's cool. So you guys aren't going to shout out the answers? I was hoping you guys would shout out the answers here, and I could just go out and copy, just write down after you. But so be it. Let's see here. What's D going to be? D is the stigma. And keep in mind, all this is female structures. All this stuff, this is all female components. So this side, female. These are all female structures. E, the whole thing is called the pistil. So the pistil is like the entirety of the female structures. Let's change colors for the male side. We got the anther, or excuse me, the anther is F. The whole structure, well, with the anther and what is the other part? The filament, I think. The filament is a part that holds up the anther, I believe. It's this right here. And keep in mind, guys, that these are the pictures you're going to see on the final. Same pictures. I didn't change it up or anything. Same exact pictures. I is the sepal. That's easy. J is the petal. And then, again, E, the whole structure is a stamen. There we go. All right, see what other questions you guys got, and then we can close it out. Oh, you guys, so the answers you guys say are the easy one. Everyone goes for petal. Easy. Easy, easy, easy. Petal. Everyone knows the petal. That's why I did it last. All right, Michael said 45 and 46. I think we're done there, right? And then we can call it a day. 45 and 46. Yeah, AB, maybe we'll go to an escape room next year. How about that? Last two, 45, 46. Oh my gosh, all these things. We label these things. Okay, we'll label these and then that's it. All right, call it a day. Okay. Left side is meiosis. All right, over here on the left. All right. The question really quick. 45, what is the structure labeled X? Where's X at? All the way up there. That's a tetrad. All right, so an allele is a version of a gene. All right, so keep that in mind. Basically, it's a type of gene. A chromatid is like one leg of the tetrad. So if a tetrad is the two X structures, or I don't know that, then a chromatid is like just one leg of the X, of one of the X's. A chromatid is just gonna be like one side. And a chromosome would be like the whole X. All right, so like the chromosome would be like just the whole structure. Whereas a chromatid would be like just one leg. So again, this is meiosis. And I did put questions in the final about like each individual phase. Like you gotta be able to recognize the different phases. And for the most part, you just gotta know which phase is which. You don't have to know like if it's interphase 1 or prophase 1 or metaphase 1. You just have to know it's metaphase or prophase. That's basically it. Don't worry about the 1s and 2s too much. So we start with, I don't think they show interphase here. I think we're going right into prophase. That's technically prophase. So I'm running out of room here, guys. Prophase 1. We got metaphase 1. Then we got anaphase 1. You tell it because we're splitting. And telophase 1. So we just put them together. Slash cyto. And then we go back to, I guess prophase. Yeah, it's going to be also right in there. Because they line up with E. So we got prophase is probably, D would probably most likely be prophase. And they probably just skipped. So we got prophase. Now we're at 2. Meta 2. You know, we call it 2 because it's the second round. And 2. And then you got telophase 2. Cytokinesis 2. So one of the main ways you can tell it's meiosis is you have 4 cells made. 1, 2, 3, 4. That should be the biggest giveaway. These 4 cells, we call them haploid. And there's a reason I mentioned all this stuff, guys. It's on the test. These are haploid cells, all right, also known as N cells. Over in mitosis, here we're only making 2 cells. 1, 2. And these are diploid cells. We would say they're 2N. So it looks like we got interphase, we got prophase, we got metaphase. Tell because they're lining up there. We got anaphase, we can tell because they're splitting apart. And then you would say like telophase and cytokinesis. Look at the numbers really quick. 1 is like, that could be the spindle. That could be the centromeres. Or no, yeah, that's going to be with the centromeres. Or excuse me, the centrioles, my bad. Those are the structures that make the spindle. So 3 is going to be the spindle. Four, that's probably just a chromosome. 2 is going to be chromatin. And chromatin is just like unorganized chromosomes. Or chromatids, you can say that too. Chromatid is the proper term. All right, let's see if you guys have any more questions. If not, that'd be it. All right, we got some stuff about Justin Bieber. Let's see here. You guys got a lot of stuff you're mentioning here. Let's see here. Can pedal be a question on the test? Maybe. You got some. I won't answer. Don't stop at my house in the summer. I won't answer the door. What questions do you have, Callie? I didn't see any of your questions. Oh, for the escape room, it's $1,000 a night, huh? Well, that's gonna be a lot of money for your, field trip fee. It's not much. Let's see here. Jeff hates you all apparently. Carter wants to know if you can say the difference between incomplete dominance and co-dominance. Okay, incomplete dominance is that's the mix. Like pink, like a pink flower and a red flower make or excuse me, a red flower and like a white flower make a pink flower. Co-dominance is they both are expressed. That'd be like if you had the same red flower and a white flower, and you get a flower that has red and white petals. So you don't mix them up. You don't get like a mix. You just get like each thing is expressed with co-dominance. I don't know what Carter wants to know about blood types. That's a lot of questions. That's like a big, broad field. You do not have to know about glycolysis. Nope, 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 nope. Hi, Gisela. How you doing? Blood type. Okay, blood type, codominance, or incomplete dominance? That's a good question. So again, incomplete dominance is a mix. Codominance is both expressed at the same time. I'm guessing that blood type would be more codominance. I'm not 100% sure about that. Ornstein Smog, yeah, that's good luck. Good luck with that. Look up a video on YouTube. Um, there we go. All right. I guess we're done here. Good job, guys. Thanks for coming. Thanks for watching this. I hope it helped. Good luck tomorrow. Get some sleep. Eat breakfast in the morning. I'll see you guys bright and early. Knock out that test, and then you're done with biology. And you just might see me next year with chemistry. Maybe. Maybe if you're very lucky. If you're very, very lucky, you just might.